Lab 2 is actually going to give you a chance to do an experiment, state a problem, form hypotheses, and design experiments for 3 different variables. And then finally collect the data on that and do a lab write-up and a lab report. So this is the best lab so far. It really goes into a lot more detail about the steps of the scientific method and how you can follow those in actually doing an experiment. So go to Content. Under Content, you're going to go down to Module 1. Now you've already done Lab 1, the data interpretation and the little exercise that we had there. So now right above that is Lab 2. There is an introduction. There are some learning objectives to take a look at. There's another introduction that I think has a pretty good TED Talk associated with it. And then there are 3 links here: how to write a lab report, introduction, how to design an experiment, and the actual experiment. Now the lab report I'd like to do last. So let's jump down here. I don't know why there are both of these steps, because the directions for doing the report are here also. But if you open this one, then the experiment is here. It's in an RTF file, and you can just download it. So in this lab, you're going to design an experiment and carry it out. Now the experiment, let's title the effect of some variable on the germination of bean seeds. The materials that you have, there are about 100 bean seeds in a pack, probably marked for this experiment. You have some plastic bags to do the experiment in, permanent marker to mark the bags. I don't know why you would need a ruler. We're not measuring how big the seeds get, we're just measuring whether they germinate or not. You need some paper towel to wrap the seeds in. You're going to keep them moist, and some masking tape to label each of the bags. Okay, let's go down to the table, and that'll tell us something about how to set this up. Table 3 says variables that may influence seed germination. Here's a column on the left to think of some variables that might affect whether some bean seeds would germinate or not. And I think the directions ask you to list 10 to 20 of those variables. It would be fine with me if you listed 10. And so let's say one of those variables is light. So light might be a variable. Does the presence or absence of light influence whether bean seeds are going to germinate or not? So our hypothesis might be that light is necessary for germination, or that light inhibits germination, or that light doesn't matter one way or another. So let's just pick one of those, like what am I doing? So let's just say that you write a hypothesis that light is necessary for bean seed germination, and do 9 more, and pick 3 that you're actually going to do experiments on. So say for the light one, you would set up the experiment in the following way. Divide your 100 beans into 3 piles, 1 for each variable experiment. For each of those piles, for each of those variable experiments, you're going to set up an experimental and a control bag. So you're going to use a total of 6 of the baggies. So 2 of the baggies, let's say you mark on them that they're for the variable of light. One of them is marked experimental, the other is the control. The experimental goes in the light, the control goes in the dark. You put about what's a third of 100? Probably 15 beans or so in each bag, so you have a pretty good sample size. And the beans, wrap in a paper towel and moisten that paper towel, put it in the baggie and put your name on them and mark where they're going to go. And you've got your setup. Now, there are 2 photos required with this lab write-up. So for photo 1, this is the beginning setup. So in your beginning setup, you would have one picture of 2 bags for light, 2 bags for the next variable that you choose, 2 bags for the next variable that you choose. All 6 bags in a row. They're all labeled with what variable they are and whether they're the experimental or the control bag. And in front of that, put a piece of paper that has your name on it and the date that you're doing this setup. And then after 7 days or 10 days, if nothing happens after 7, then you do the same thing. I'd like to see a picture again of those 6 bags in a row that is clear enough and close enough that I can see where seeds have germinated and if there are any bags where the seeds have not germinated. So those 2 photos. Again, on this second photo, you have a piece of paper in front of it that has your name on it and the date. When you have your results, create some tables for your data, or one table for your data, that includes the results of all 3 labs, and then a graph of that data. So a data table would be like this. I don't know why this is labeled table 3, but so name your data table table 4, and then the effect of variables on the germination of bean seeds. And construct it so that you can record your data any way that you like there. And then make a graph of that data. Now, you could make 3 different graphs, but you don't have to. It seems that this data is going to be discrete data. How many in the experimental light germinated? How many in the control light germinated? And then same experimental control for your other 2 variables. That's not continuous data, so a line graph probably isn't appropriate. So it's a bar graph. And in that bar graph, you could have 2 bars for your first variable, 2 bars experimental and control for your second variable, 2 for the third variable. The independent variable is I have to stop and think about this because I always mix these up. The independent variable is the x-axis, and it is the condition that the seeds were in. So variable 1, variable 2, variable 3, experimental and control for each one. The dependent variable is how many seeds actually germinated in each of those. So your y-axis is going to be number of seeds, and construct the numbers on that so that you do get some difference in the bars, that they're not all either right up at the top together or right down at the bottom together. A graph is a figure, and figure number and titles go at the bottom. Okay, so you've done your 3 experiments, you've got all the data collected, you have a nice data table. This is one of the data tables. Data table 4 has your results in it, and figure number 1 is your graph of that data. Now let's go back and see how to do the lab write-up. We're back to Content Module 1, and we're working down here on Lab 2. How to do the lab write-up is this link. Open that. How to do the lab report. There's some introduction about following the scientific method and what kind of data you refute or whether you have to publish, things like that we've already talked about. All right, the format for the report, 1 Word document or 1 RTF document. The first thing in the lab report is the title. Title's the effect of 3 different variables on the germination of bean seedlings. The abstract is a summary of your whole experiment. It's something that's shorter, that if this were really a research paper that was published, other scholars would want to come and see what you did, but they don't want to read through the whole experiment. So they want to just read a brief summary or an abstract of it. So after you do the title, put the heading for abstract there, but you actually don't write the abstract until the very end. The next section is the introduction. It's an overview of why the experiment was conducted and how you did it. Background, objective, hypotheses. You don't have to label each of these, but you can if you want. Going to tell why you did the experiment, some background information about bean seeds. I guess I'd expect the background to be about germination in general. What is germination? What do seeds do when they germinate? And something about environmental variables. The objective, what your 3 hypotheses were for the 3 different experiments. Next is material and methods. Just describe how you did the experiment. It would be in the past tense because you've already done it. Describe it in enough detail in 1 descriptive paragraph, hopefully. Not too long, just how you did it, that somebody else who's coming along could follow along and do it too. Then your results section, describe what happened in each of the 3 experiments. Describe that in another descriptive paragraph, and then at the very end of your description, say see Table 3, Table 4, and Figure 1. And then right after that, you're going to have the table with all your variables, the 10 variables. You're going to have the next table that is your results, and you're going to have Figure 1, which is the graph, all under this results section. And then follow that with your photos. And now the photos, I don't care. They could be at the beginning of the results, they could be at the end of the results, but they are showing what happened in your experiment, so they should be in this section also. And then finally, a discussion. This is where you tell why the results came out the way they did. How do you interpret the results? And then this direction is asking for a conclusion. Just summarize what you said in the other sections in very short. I don't even care if you leave this conclusion out, really. And then if you cited anything, if you looked anywhere, like for your background about germination, then do a reference here and an in-text citation where you use that. After you do all of that, then go back up and think of doing a piece of introduction, a piece of method, a piece of results, and a piece of discussion as a final summary. I wouldn't think over, I don't know, 75 words or no more than 100 words. It says not to exceed 200, but you could be a lot shorter with that. And so that's the abstract, goes in last, just summarizes the whole experiment. Okay, that's it. So a good experiment, interesting experiment. Hope you think of 3 really good variables to do that are easy to experiment with. Do a nice report, put it in the Dropbox before the final due date. Look at that final due date. Give yourselves a couple days to write it up, and then back up about 10 days to begin. And I guess looking at that, I would think you'd want to begin this lab by Thursday or Friday, but no later than Saturday of this week for sure. Good luck. Let me know if you have any questions.